Hello and welcome to True Pathfinder, your portal to health and truth. Alzheimer's disease and other types of dementia are truly devastating, and it's extremely hard to witness how your loved ones slip away into those conditions. And the sad part about it is that despite billions and billions of dollars that have been put into research on this subject over the years, we still cannot cure this disease, or mainstream medicine cannot, not even slow it down for that matter. But there is some good news, because there seems to be a recent breakthrough, some amazing new research that explains why we have not found a cure yet. And this has to do with parasites, as the title suggests. So let's look at these amazing new results and what that means for a solution to this debilitating disease. Alzheimer has been continuously on the rise over the past decades. It affects 5.3 million Americans and is the sixth leading cause of death in the US. It is also the only one of the top 10 killers that, according to mainstream medicine, cannot be prevented, cured, or slowed down. In 2015, Alzheimer's and other types of dementia cost the US $226 billion, and by 2050 this cost could rise to 1.1 trillion, according to the Alzheimer's Association. The disease is characterized by plaques in the brain, which form when sticky protein pieces called beta amyloids clump together. Despite billions being invested to understand how those beta amyloids contribute to a slow cognitive decline, it remains a mystery. And this lack of progress in the understanding and treatment of Alzheimer's suggests that the etiological hypotheses followed by the mainstream scientific community are either wrong or at least incomplete. We truly need a paradigm shift, and such a shift has been quietly but consistently taking place over the past years. Let's look at those novel discoveries and the new understanding they provide for the causes and treatment opportunities of different types of dementia and Alzheimer's. First, let's look at what Alzheimer is. It is the most frequent cause of dementia and is characterized by a slowly progressive decline of cognition and memory. Alois Alzheimer, the discoverer of the condition, described the characteristic cortical senile plaques and neurofibrillary tangles in the brain of a 51-year-old woman who suffered from presenile dementia, and you can see her here in the photograph on the right. Her name was Auguste, and she is the first documented case of Alzheimer's disease. And recently it was pointed out that the presenile form, which onsets before the age of 65, is identical to the most common form of senile dementia, and therefore today the term Alzheimer's disease is used for the designation of both presenile and senile cases. The pathological hallmarks of Alzheimer's are a progressive brain atrophy and the accumulation of cortical senile plaques, as well as neurofibrillary tangles. A fibrillary amyloid substance is deposited in the senile plaques formed by the aggregation of the 4.2-kD amyloid beta peptide, an A-beta amyloid, and granulosing degeneration is another characteristic alteration of neurons in Alzheimer patients. The origins of are beta deposition. the neuronal tangle formation and the granulovacuolar degeneration, still remain unclear despite various hypotheses that have been brought forward over the years. One thing is clear though, chronic inflammation plays a critical role in Alzheimer's disease. The question is only, what causes this chronic inflammation? It is very important to note that the recognition that pathogens can produce slowly progressive chronic diseases has resulted in a new concept of many such chronic conditions over the past decades. For example, the pioneering work of Marshall and Warren established that Helicobacter pylori causes stomach ulcers. The etiologic agent of Whipple's disease was revealed to be another bacterium called Treponema Whippleyi. Recent reports have also documented that infectious agents also occur in atherosclerosis, cardio and cerebrovascular disorders, diabetes mellitus, chronic lung and inflammatory bowel diseases, as well as various neurological and neuropsychiatric disorders. Interestingly, nearly a century ago, Fisher and Alzheimer himself, together with their colleagues, already discussed the possibility that microorganisms may play a role in the formation of senile plaques. Historic data indicate that the clinical and pathological hallmarks of syphilitic dementia in the atrophic form of general paresis, which is a type of paralysis, caused by chronic spirochetal infection, are similar to those of Alzheimer's disease. And by the way, I also highly recommend, we've done a lot of videos on these microparasites that cause chronic conditions, and we've showed you live footage in one of our videos of these microparasites in cancer patients. It's actually the most viewed video that we've ever produced with hundreds of thousands of views. I will post it down below. Highly recommended that you take a look at that as well. Alright then, let's look at this breakthrough new recent research and what it has revealed when it comes to Alzheimer's disease. But before we do that, short reminder, if you enjoy our content, please support us directly by becoming a patron from as little as $5 per month. Link is in the description box below. Also, do not forget to like, share, subscribe if you're not already, and comment to help this channel grow and spread the truth. For the holistic care you need and deserve, please contact us at christine.doctor. We can assess you remotely wherever you are in the world and will develop a holistic natural healing plan that is customized to your needs. We have a lot of experience with all of these microparasites and the chronic diseases they cause, such as Lyme disease, so do not hesitate. Contact us and we will help you. Also, check out some of the most powerful holistic health tools available on the market today at vibrantbody.net. Some of them can also help you with those microparasites that cause many chronic conditions. But back to Alzheimer's. In the early 90s, pathologist Alan MacDonald theorized that Alzheimer's disease may have a bacterial component. And later, Swiss researcher Judith McCloskey made a profound observation when she showed that neurosyphilis, which is a bacterial disease of the brain, and Alzheimer's were essentially identical. McCloskey and her colleagues found two types of spirochetes, which are long, corkscrew-shaped bacteria in the brains of more than 90% of Alzheimer's disease patients. Those spirochetes were Borrelia burgdorferi, which of course, as you might know, causes Lyme disease, with which we have a lot of experience, and periodontal pathogens that are close relatives of syphilis. Studies from other researchers have made similar connections between bacteria and dementia. The findings also intrigued Dr. Herbert Allen, who has decades of experience in the treatment of patients with both Lyme disease and syphilis. And in a 2014 study, Dr. Allen showed that eczema may be caused by biofilms. What are biofilms? They are multicellular assemblages of bacteria that exude slimy glue-like substances. Once the bacteria metamorphose into a biofilm, antibiotics can no longer attack them. The 2014 study proposed that staphylococcal bacteria that live on the skin produce also a biofilm that blocks the sweat ducts, activating the immune system, which in return causes itching and irritation. Dr. Allen's newest study, published in Neuroinfectious Diseases, suggests that spirochetes in the brain could be operating in the same way to cause Alzheimer's disease and other types of dementia for that matter. Dr. Allen and a team of scientists investigated 7 post-mortem brains of Alzheimer's disease patients, comparing them to 10 control samples from people who had died from other causes. And amazingly, in each one of the Alzheimer's brains, the researchers saw what they believed to be biofilms. They also identified a protein called TLR2, which plays a key role in activating the body's innate immune system, which hints again to the fact that there is an inflammation in those people's brains. According to Dr. Allen's hypothesis, the spirochetes enter the brain during a dental procedure, for example, or after a person contracts Lyme disease, and then spin out a protective biofilm. The body's first responders try to clear the infection, but immune cells cannot penetrate the biofilm matrix that encases the organisms. So what does the immune system instead ends up doing? It destroys the surrounding tissue. This is, by the way, also exactly what happens in cystic fibrosis. As for why Alzheimer's primarily strikes older adults, the study's authors note that spirochetes take 90 days to divide. So the process to create this biofilm takes a very long time. It takes a long time to gain sufficient numbers of organisms to form it. However, interestingly, after someone suffers a stroke, the adaptive immune system, which the authors liken to a machine gun, is activated and eradicates organisms with much greater efficiency than the innate immune system. Quote from Dr. Allen and his colleagues, however, just as in the situation with the innate immune system, it cannot penetrate the biofilm either, thus it destroys great amounts of the surrounding neural tissue much more rapidly. The hypothesis could also explain why symptoms of dementia can appear suddenly after a stroke rather than slowly over time, as is observed with traditional Alzheimer's patients. Dr. Alan MacDonald, Dr. Allen, and other of these pioneering researchers have performed a whole range of different tests to prove that these microparasites and the biofilm they create can be found in almost all Alzheimer disease patients. Here you see some fascinating picture from one of the studies by Dr. MacDonald. It's a microscopic study of 100 consecutive Alzheimer disease amyloid plaques, and they demonstrated that 100% of them contained those biofilms of Borrelia dwelling inside of the topographies of each of the amyloid plaques. On the left you see the granular forms of the Borrelia in the biofilm. On the right you see again the autopsy pictures from the brain of the Alzheimer disease patients stained in different ways, and the conclusion is the Borrelia, in this case the Nematodes strain DNA, is abundant inside of the amyloid plaque in the Alzheimer disease patients, and they use here the fluorescence in situ DNA hybridization method, and they used other methods as well, as we will see, always with the same result. In the picture here on the left, in the A sample, you see Alzheimer amyloid plaque stained with thioflavin T stain. Amyloid is bright green, and note that the voids inside of the green plaque do not stain for amyloid, and these are present, as you can see, in image A and B, and are characteristic of the empty spaces inside of amyloid Alzheimer's plaques. In picture B on the left, we see Alzheimer amyloid plaque stained with Borrelia Spirochetes, and note that here the voids inside of the plaque are again demonstrated as in figure A, and they are present throughout the plaque. These are not amyloid, but rather the granular Borrelia forms, which are very characteristic of the closely packed specialized Borrelia Spirochetes forms in a biofilm community, according to the researchers. On the right, you see again a thioflavin T stained plaque, and the DNA probe is stained with the Borrelia Spirochetes. In this plane view, you cannot see the water channels, which are used in the biofilm by the bacteria to secrete, to purge out the debris and the remnants that they don't need anymore, and also to nourish themselves. But in other pictures that they've taken of these biofilms, the water channels can also be clearly seen. Borrelia Spirochetes were not the only types found in Alzheimer disease patients. They also found Treponema Spirochetes. This study was done by Dr. Mikosi from Switzerland, and the title of the study is Alzheimer's Disease, Neurospirochetosis, Analysis of the Evidence Following Koch's and Hill's Criteria. And in the first figure here, you can see the association of the spirochetes with Alzheimer's disease. They used the control group and the Alzheimer's disease patients, of course, and they say that the frequency of spirochetes is significantly higher in the brains of Alzheimer patients compared to controls. The statistical analysis is based on the cumulative data of the literature entered in Table 1, and the association is statistically significant in the 4 groups analyzed. In the group where all types of spirochetes were detected using neutral techniques, in the group of only oral periodontal pathogen spirochetes, and in the group where Borrelia burgdorferi was detected alone, and also in the group where all studies were considered. As you can see from the bars here in the graph as well in the visual representation, whether you look at all spirochetes or only certain types, there is always much higher concentration to be found in Alzheimer's disease patient brains. In the graph here on the right, you see the association of oral invasive periodontal Treponema Spirochetes with Alzheimer's disease, and using species-specific PCR and antibodies, 6 of 7 periodontal pathogen spirochetes analyzed were detected in the brains of Alzheimer's disease patients. So the researchers found all of these types of Treponema spirochetes in the patients' brains, and they also say that the association of these oral treponemas with Alzheimer's disease is statistically significant. And believe it or not, all of these different types of spirochetes were not the only microparasites that were found in the Alzheimer patients' brains. They also found parasitic nematode worms. Here in the picture from a study by Dr. McDonald from 2016, we see the Alzheimer's disease brain with two nematode worm parasites. The trichrome stain, so the blue and red colors, was used here to highlight the worms, and the small particles of blue and red demonstrate worm debris, which is released from the nematodes as they migrate through the Alzheimer's brain. The brain itself does not stain with this trichrome stain and is therefore nearly colorless. Particles of the worm debris are refractile, and intact worms are also refractile, and refractile materials stand out therefore from the healthy brain tissue, which is always non-refractile in photomicrographs. On the right here you see another image of an Alzheimer's disease brain that was autopsied, and here we can observe two Alzheimer's plaques which are stained with acridine orange stain. The edges of the plaques are sharp and distinct, as you can see, and the plaques are the biofilm communities that we have discussed. We see living microbes that are present in the biofilm. We also see the protective matrix material that surrounds the entire biofilm community. And we also see the empty spaces that percolate through the biofilm plaques. And these are the water channels that we've mentioned already, which provide nutrition to the biofilm, and also a sewer-like system for waste product removal. We also see that the particles in the biofilm plaques include red and refractile nematode worm debris and minute non-refractile microbes. Time for conclusions then. As we have seen, all Alzheimer's plaques contained amyloid plaques, as expected. But, as not expected, they also contain biofilms of different species of spirochetes, such as Borrelia and Treponema. And other parasites, as we have seen, were found as well in Alzheimer's disease brains, such as the nematodes. Biofilms are a typical survival tactic that is used by many types of bacteria. 99% of them use this biofilm tactic. In tissues, biofilm formation always indicates an inflammation, a chronic infection, and biofilms grow slowly over time. As we age, these living biofilm communities grow in size and increase in complexity of architecture. And these results are consistent with other findings like the microparasites found in all cancer patients. Again, I highly recommend that you watch this video that we did on the subject. I'm gonna link it down below. These findings could shift the paradigm of Alzheimer's and other types of dementia disease research. They could mean that rather than trying to develop drugs to try and target the beta amyloid, as is the direction of almost all current studies, researchers could instead be putting efforts into developing more reliable tests for bacterial detection in the central nervous system and investigating new anti-biofilm treatments. We know that a healthy terrain or milieu prevents chronic conditions. So, take care of your lifestyle, nutrition, exercise, stress levels, etc., etc. Also, do not neglect your mental and psychological health. And get the right holistic, preferably natural, therapy. We can help you with that here at christine.doctor. Please contact us. We know of a whole range of very efficient natural microparasitic killers or pathogen killers that have been proven over the years to work for many types of chronic conditions. Link is, as mentioned, in the description box below. Also check out some of the most powerful holistic health tools at vibranbody.net. Some of them can help you also greatly with the eradication of these pathogens. If you've enjoyed this presentation, please support us directly by becoming a patron from as little as $5 per month. Link is of course also in the description box below. And do not forget to like, share, subscribe if you're not already, and comment to help the channel grow and spread the truth. With all it being said, stay healthy. Until next time.